Hello everyone, my name is Charlotte and I will be your host for today. Welcome and thank you for taking part in MDPI's webinar entitled Nutrigenetics, Translation into Clinical and Nutritional Practice. This is the third webinar in the nutrient series based on special issues from our journal of the same name. If you are interested in contributing to this topic, check out the webinar's webpage for publication opportunities in the journal's special issues. I would like to remind everyone that this webinar is being recorded and the recording will be made available as soon as possible on the webinar's webpage. Please note that you will have to be logged into SciForum in order to view the recording. Today's session is also being live streamed on MDPI's YouTube channel, so hello to those of you watching us on there. Certificates of attendance are available upon request. To get yours, please send an email to nutrients.webinar@mdpi.com. Certificates will be delivered to those who have registered and are watching on Zoom, not on YouTube. Thank you for your understanding. There will be 3 presentations today of 40 minutes each. The presentations are followed by a question time moderated by the chair. If you have any questions for our speakers, please post them in the Q&A box at the bottom of the screen. Please remember to mention the name of the person to whom you're addressing your question. Today's webinar is chaired by Professor Dr. María Luisa Bonet from the Nutrigenomics and Obesity Research Group at the Laboratory of Molecular Biology at the University of the Balearic Islands. María Luisa is a professor of biochemistry and molecular biology and also co-director of the Master in Nutrigenomics and Personalized Nutrition at the University of the Balearic Islands, UIB. Her background is in molecular nutrition, nutrigenomics, and the physiopathology of obesity. Her research relates to the mechanisms regulating body weight and adiposity in mammals and their interaction with dietary factors, notably vitamin A-related compounds, including the impact of early life nutrition in the long-term programming of the susceptibility to obesity and related health outcomes. Thank you very much, María Luisa, for chairing today's webinar. That's it from me. I now hand over the microphone to our chair and wish you all a pleasant webinar.
Okay, thank you very much, Charlotte, for this kind introduction. And I will do some introduction to the seminar myself. Welcome to the Nutrients webinar, Nutrigenetics Translation into Clinical and Nutritional Practice. This seminar is an activity organized from the Nutrigenetics and Nutrigenomics section of the journal Nutrients. This section of Nutrients was created in 2020 with the aim to publish contributions on all aspects of nutrigenetics and nutrigenomics, in particular contributions that provide novel and mechanistic insights and constitute significant advance in the fields. As a short introduction to the topic, we know that there are multiple biological layers of variability among individuals that affect the relationship between diet and health, and that are themselves affected or programmed by nutritional conditions of life. Among them, genome, epigenome, the gut microbiome, health conditions, lifestyle factors. And today we will center mainly in the impact of genome variability, which is the field of nutrigenetics. Okay. This slide shows that genetic variability affecting the impact of diet on the individual's performance and health status can impact different biological functions. So single nucleotide polymorphism and other types of genetic variation may affect, for instance, dietary choices and food intake and behavior. And there are also genetic variation that can affect the bioavailability and metabolism of nutrients and other food bioactives, including SNPs that can affect host gut microbiota composition and quantity. And finally, there are also SNPs that can influence the metabolic responses of our tissues to nutrients and diets. And we are in these recent years beginning to grasp this genetic variability in the human genome and its implications for health, behavior, capacities, both physical and intellectual capacities, the way we age, and responses to different types of interventions. Even if still in its infancy, the development of nutrigenetics and nutrigenomics is already providing concrete examples of interactions of genetics, nutrition, and health outcomes, whose translation into the clinical and nutritional practice begins to be feasible and advisable. This knowledge can be used in preventive and therapeutic approaches for better health and performance through personalized dietary recommendations. And moreover, at the same time, this novel knowledge is increasing our understanding of human biology and physiology. Well, so in this context, as scientific organizer and chair of this session, it is my pleasure and my honor to have reunited in this webinar three of the most outstanding scientists acknowledged worldwide in the field of nutrigenetics. Professor María Minihane from University of East Anglia, UK, Professor Alfredo Martínez from University of Navarra, Ibero-BN, and INDEA in Madrid, Spain, and Professor Ahmed El-Soemi from the University of Toronto, who will, these speakers, relevant speakers, will show us relevant examples emerging from their research and also the momentum of the area. Now, after this short presentation, let me just introduce our first speaker, which is Professor Anne-Marie Minihane, from the University of East Anglia, Norwich Medical School in the United Kingdom. Anne-Marie Minihane is Professor of Nutrigenetics, Head of Nutrition and Preventive Medicine in Norwich Medical School, and Director of Norfolk Institute of Healthy Aging. Her research investigates the impact of select dietary components, in particular omega-3 fatty acids, and a Mediterranean-style dietary pattern on cardiometabolic and cognitive health. She also investigated nutrient genotype interactions with a focus on the APOE genotype. And in fact, her presentations today will be about APOE genotype, aging, brain health, and the response to dietary change. So, Anne-Marie, with 2 minutes behind a schedule, but the floor is yours. Thank you.
Louisa, thank you very much for those kind words, and thank you for the invite to speak here today. Good afternoon, everyone from Norwich, UK, and also thank you, Charlotte, for organising this so slickly. Much appreciated. So, as Louisa said, today I will speak to you about a topic I've been working on now for over 20 years, which is the role of APOE genotype in health and response to dietary change. And actually, I just checked this morning, if you look in PubMed, there's 10,518 papers which have a focus on APOE genotype and health in response to dietary change. So it's a much researched area. I thought I'd start off by just giving a bit of background, as I'm the first presentation, on types of genetic variation and the concepts of nutrigenetics, and then go on to talk about what APOE genotype is, how it's related to health endpoints, what we know about how it modulates response to diet, and just some final thoughts about, this move from generic to personalization and how that might all work and how far along we are. So this is what, in my mind, the genetic discovery timeline looks like. So we've known actually since the middle of the 1800s, from work by Charles Darwin and Alfred Wallace and Gregor Mendel, that our genetics interacts with our environment to determine phenotype. Now, Mendel was interested in peas, but of course the phenotype we're quite interested in today is health and well-being. But it wasn't until the 1950s that the structure of DNA was discovered by Watson Crick and Rosalind Franklin. But it's actually even much more recent than that, and only over the last 20 years or so, that we've begun to appreciate the structure of the genome and the amount of variability in the genome. And we now know the genome is made up of about 22,000 genes, 3 billion base pairs, and somewhere in the order of 88 to 100 million variants. And of course, that's a lot to get a handle on. And when all of this was discovered, it was considered the panacea, and that it would really revolutionize our ability to diagnose disease, or diagnose disease risk, and really revolutionize our ability to prevent and treat disease. But of course, a lot of that potential is yet to be realized, but there's some good examples coming through, and I have to say APOE is one of those. So just a bit about genetic variability. We're all almost completely genetically identical, 99%, 99.9% identical, and 0.1% variable. And that variability can take the form of large structural alterations to our DNA, and that constitutes less than 10% of total genetic variability. And it underpins about 10,000 monogenic disorders, things like sickle cell anemia, Huntington's disease, some forms of familial hypercholesteremia, some forms of early onset AD, Alzheimer's disease, etc. But from a public health perspective, we're more interested in the other 90%, which is common genetic variants of the single nucleotide polymorphism variety. And as the name suggests, this affects just a single nucleotide in the DNA. And that nucleotide change can result in it can have no effect on the protein amino acid sequence, and it's referred to as synonymous. If it affects the amino acid sequence of the protein, it's called non-synonymous. And if it results in a shortened protein, a nonsense SNP can result in a shortened protein, which can also have a large effect on protein function. And these type of genetic variations, SNPs, contribute to polygenic disease risk, where there's a number of variants contributing to the same disease. And of course, we know, unlike monogenic disorders, these are predictive rather than fatalistic. So if you have the variant, you may or may not develop the condition, depending on lots of other variables. And when we look at that variation and its impact on risk of disease and response to environmental factors, it's mediated in very general terms by affecting either the expression of the gene or else the protein structure, or where the protein is localized in a tissue or cell, or the stability of the protein. So the variant can affect risk of disease and response to diet in a number of different ways. And Luisa gave a very nice overview of nutrigenetics in one of her opening slides, and this is just to reiterate this. When we talk about our genotype interacting with our diet to affect health status, it can work at a number of levels. So our genotype can affect our food intake and our preference. Once we eat a particular food, it can affect the absorption and distribution of dietary components. For particular nutrients, such as vitamin D, it can affect our ability to endogenously synthesize the nutrient of interest. And once you reach a particular nutrition status, its physiological impact and its effect on health can very much be determined by genotype. So nutrigenetics can work at a number of different levels. And I often get asked, will this kind of personalized approach to nutrition replace the more generic population dietary guidelines? And I don't see that being the case at all. I think more personalized approaches based on things like genetics will just accompany and enhance the more generic population recommendations. But there are two main reasons in my mind why we should think about more personalized approaches. The first thing is, at a population level, often adherence to our more generic dietary recommendations is poor. And also at a population level, responsiveness to dietary change is hugely variable. So these two facts kind of push us in the direction of trying to get a bit more personalized in our approaches. So I look at these two things separately, and number one is just at a population level, adherence to dietary recommendations is often poor. And this was actually a paper published last year based in the UK. And it looked at how well we complied to our Eat Well plate. And if you have the perfect Eat Well plate, it can be characterized by 9 different food or dietary components. And what this graph shows here was that 70% of the population only adhered to 4 or less of those dietary recommendations. And actually 40% of the population only adhered to 3 of the 9 Eat Well recommendations. And when we look at individual dietary recommendations, again, the picture was quite poor. So only 7% of the UK population met the fibre recommendation, only 26% of the population met the non-oily fish recommendation. So adherence in general is poor to dietary recommendations. The second thing is that a population level response to dietary change is hugely variable. And this is data from an old study of ours called the FENGEN study, which involved 312 participants. It was a crossover study, which was looking at the ability of fish oils to positively modulate the plasma lipid response. And this graph here shows the triglyceride response to fish oil supplementation over 8 weeks. And at a group level, we saw a very nice 11% reduction in plasma triglyceride levels. But this is each of the 312 participants plotted individually. And as you can see, the response is hugely variable. And actually, this participant, R245, had a beautiful 250% reduction, but R109 actually had 150% increase. And actually, overall, 118 of the 312 participants experienced an increase in triglycerides, even though the overall group change was positive and an 11% reduction, showing this variability in response, which we need to get a handle on. So today I'm talking about APOE. As I say, there's a huge number of publications on APOE already, but we haven't quite nailed it yet. APOE gene is on chromosome 19. APOE as a protein is mainly produced by the liver, but also significant amounts produced by the brain and macrophages. It's quite a key modulator of the immune response. And there are 2 particular SNPs, RS7412 and RS429358, which affect the amino acid sequence of the protein, producing 3 versions of the protein, E2, E3, and E4. And of course, we all have 2 copies of each allele, so we can have one of those 6 genotypes listed on the bottom, going from E2, E2 to E4, E4, with E3, E3 being the common wild-type genotype, and E4 carriers considered to be the risky genotype. APOE4 genotype in particular affects the orientation of the protein and creates a salt bridge which affects the interaction between the C and the N terminal of the protein, which really affects the functionality of the protein and also really affects the stability of the protein. And as you can see, these are plasma APOE levels. As you go from E2, E2 to E4, E4, you see a quite dramatic decline in the concentration of APOE in plasma and also in tissues. Going on to the health effects of APOE. APOE has emerged as one of the most significant determinants of longevity and lifespan. It's estimated that about 25% of longevity is determined by genetic factors. And if we look at this particular publication, which is quite an old one now, which was published in 2006, it accumulated data from 12 independent studies. And what it did is it looked at the APOE allele frequency at different ages. It looked at E2, E3, and E4. And as you can see in each of the 12 studies, as the population got older, the frequency of the APOE4 allele decreased, which was indicating that people who were E4 were beginning to die off. And associated with that was a compensatory increase in the E3 and the E2 allele. This is another study looking at APOE and longevity. called the Genetics of Healthy Aging study, where they took people in their 90s and compared them to people between the ages of 55 to 75. And what they found was the allele frequency of APOE4 was about 7% for a group in their 90s, but was much higher, about 13% between the ages of 55 to 75. And actually the E4 allele reduced your chance by 50% of making it to the age of 90 or above, which is quite a dramatic effect, I have to say. And that finding very much concurs with a more recent meta-analysis, which was produced in 2019 in Nature Communications. This was a very elegant analysis, and it took data from a whole range of discovery cohorts. And you can see the GHAS is the analysis I was talking about in the previous slide. So it looked at 18 separate discovery cohorts and 2 replication cohorts and a validation cohort. And it looked at people who had made it into the 90th or the 99th percentile of life. And it compared those to people, the control group, who had whose age at death or the last contact was less than the 60th survival percentile. So these were much younger folks. And they looked to see what genetic determinants predicted which group people were in. And this is a bit of a complicated slide, but what I'll draw your attention to here is this particular genotype, 429358, which is APOE4, and the 7412, which is APOE2. And you can see, concurring with the previous analysis, if you're APOE4, it reduced your risk of being in the 90th percentile of life by about 55%. But if you were APOE2, you were 32% more likely of making it into the 90th percentile. And the odds ratios were similar when you looked at the 99th percentile, against strong effect sizes of APOE on longevity and lifespan. Now, what's underpinning this is the big question. And the first thing that's underpinning this is the fact that if you're APOE4, you're at increased cardiovascular risk. And this is just a slide summarizing a number of studies, and it looks at the odds ratio of cardiovascular disease if you're APOE4. And as you can see, in almost all studies, being APOE4 increases your risk by various degrees, ranging from about 6% in this study to about 66% in this 2005 analysis. And what's the physiological basis for that? And it's thought to be multiple-fold, actually. If you're APOE4, it tends to detrimentally affect your plasma lipid levels. You have an exaggerated postprandial lipemia. You tend to have an exaggerated inflammatory response and also be in a heightened state of oxidative stress. And I'll just show you a little bit of the data to demonstrate this. And this slide looks at the association between APOE genotype and LDL cholesterol. And you see a very, very clear dose response when you go from E2, E2 to E4, E4, with the E4, E4s having about a 20% higher LDL cholesterol relative to the wild type E3, E3 genotype. The next slide is some data from our own group, which we published a while ago now, which looked at the effects of APOE genotype on postprandial lipemia, our ability to metabolize fat after a high-fat meal. In this group of people, we gave a breakfast and a lunch containing 49 grams of fat or 29 grams of fat, and we took regular blood samples. And as you can see, E4 carriers had both an exaggerated fasting triglyceride response, but also had a much heightened postprandial lipemic response. So again, kind of demonstrating that this is a basis for the increased cardiovascular risk. One of the other main reasons, or probably the main reason why APOE4s have a shorter lifespan, is it increases your risk of dementia. Dementia is now the main source of death in the UK. About 13% of us die of dementia, actually much higher in women than in men. 2/3 of dementia cases are women, And it's not just due to, as shown in this figure on the right, it's not just due to the fact that women live slightly longer, but women also have a higher age standardized prevalence of dementia. This slide shows here the effects of APOE genotype on dementia risk. This is the common genotype here in pink, the E3E3, and we use that as the reference. On average, somebody who's E3E3 develops dementia at the age of 84. If you're E3E4 or an E4 carrier, you have a 4-fold increased risk, and it really results in an earlier age of onset, 76 years on average. If you happen to be double E4, which is about 1 to 2% of the population, your relative risk is 16-fold higher, and your age of onset is almost 20 years earlier. So a major determinant of Alzheimer's disease risk. Interestingly, the penetrance of APOE genotype on risk seems to have a large ethnic underpinning basis. So this just shows 4 different ancestral backgrounds here: Caucasians, African Americans, Hispanics, and Japanese. And if you look at the effects of the 4E4 genotype on risk, you can see in Caucasians it's about somewhere between 10 and 15-fold increased risk. That's much reduced in African Americans. It's almost completely absent in Hispanics, but it's much more even exaggerated in the Japanese population, which kind of demonstrate that the penetrance of genotype is influenced by a whole host of other factors, including things like diet. Also, something we've got really interested in is that the penetrance of genotype seems to be really affected by your sex. And this is really interesting, and it's just starting to come to light. Now, this is a bit of a complicated graph, but the 2 bits of data lined on top of each other are comparing men and women. And the overall reference point is the E3E3 group. So I'll just explain that a bit better now. If you go over here, healthy control to Alzheimer's disease, mild cognitive impairment, or Alzheimer's disease conversion, the orange is female and the blue is male. And what this is showing is relative to the E3E3 genotype. If you're E3E4 and you're female versus male, you have a higher chance of making that conversion between a healthy person to an Alzheimer's disease person. And if you look at another pairing over here, this just looks at Alzheimer's disease risk in E3E4s compared to E3E3, whether you're female or male. And again, you can see that being female and APOE4 is associated with a greater risk of Alzheimer's disease compared to being E4. So yeah, being an E4 carrier is much more consequential for Alzheimer's disease risk or dementia risk if you're female rather than male. And we're just starting to try and figure out what the mechanistic basis for that is. Now, when you look at the effects of APOE genotype on Alzheimer's disease risk, APOE and APOE4 does a whole host of things, and I'll just focus on 2 of these today. One we're really interested in ourselves, and the second one I'll start off with, which is the effects of being APOE4 on amyloid beta deposition. And of course, amyloid beta deposition is one of the defining features of the Alzheimer's disease form of dementia. And what this graph shows here is it divides people by age, and it looks at whether you're an E4 carrier or a non-E4 carrier. And it's looking at PET scans, and it's looking at PIB positive. And PIB positive reflects basically amyloid accumulation in the brain. And as you can see, at every age, but particularly as you get older, there's a much higher accumulation of amyloid beta in the brain in E4 carriers versus non-E4 carriers. We're very interested on the effects of APOE genotype on omega-3 status in the brain. And the reason for that is as follows. If you look at different tissues in the body, going from adipose tissue to heart to muscle to the brain, the cerebral cortex, you can see that the brain is particularly enriched in the omega-3 fatty acid DHA, where it makes up about 15% of total lipid in the brain, compared to less than 2% in tissues like the heart and the liver, which is kind of indicative of the importance of DHA in the brain. And it's even more enriched in synaptic regions where neurons talk to each other. And there's a lot of prospective epidemiological evidence to show that increased intakes of DHA is associated with higher total and hippocampal brain volume, better cognitive performance, reduced risk of Alzheimer's disease. And this is supported by a whole host of very convincing experimental model evidence. So we started to get interested in, does APOE genotype influence DHA levels in the brain, and is that mediated by sex? And this was a very simple experiment we did actually, where we took rodents, which were mice, which were either E3 or E4. They had been stably transfected with either the human APOE3 or the human APOE4 gene. And we aged them to different ages, and we sacrificed them at 2 months, 9 months, and 18 months. And as you can see, as the animals aged, there was a decline in DHA levels in the brain, but this was much more significant in females, and it was much more significant in E4 animals. This made us start to think about a little bit about the effects of menopause on cognition. Is some of this decline in females somehow modulated by menopause? So with this in mind, we have just recently completed a series of rodent studies, again in E3 and E4 animals, where we induced menopause. So we fed the animals on a high-fat diet for a period of time, and then we induced menopause in these animals with a compound called VCD. And what VCD does is it targets the ovarian follicles which produce estrogen. So the animals produce a very human-like menopause. And then we looked at the effects of the induction of menopause in these animals according to APOE genotype. And we looked at cognition, we also looked at brain DHA levels, and we also looked at some neurophysiological targets. And what we found was that genotype actually reduced spatial memory in these animals, but it was unaffected by the menopause. But interestingly, when you look at recognition memory, which is a form of episodic memory, E4 animals were particularly susceptible to the effects of menopause, where we saw a sharp decline in recognition memory in E4 animals only following the induction of menopause. We looked at levels of DHA in the brain, and we found actually that menopause reduced DHA levels in both genotype groups. These animals were actually sacrificed at 12 months rather than 18 months in the previous study. And we actually saw that some of the receptors which modulate uptake of DHA in the brain were also beginning to be affected by both genotype and the induction of menopause. And actually, this next slide is hugely complicated, and don't worry about it too much. But we looked at synaptic function as a molecular target, the synaptic region of neurons. And we looked at markers like BDNF, mTOR, things like mTOR, AKT, Krebs signaling. And we found that both APOE4 and menopause detrimentally affected a whole host of synaptic function related pathways in these animals. And actually, interestingly, we did a follow-up study, which we've not published yet, where we actually supplemented the animals with DHA. And we found that when it comes to both brain DHA levels, this is the supplemented animals here in green, when it comes to both brain DHA levels and also cognition, that actually supplementing with DHA rescued the phenotype. So what all this means actually is that APOE4 carrier status is associated with reduced longevity, increased cardiovascular risk, and increased dementia risk with APOE4s are women being particularly susceptible to the effects of APOE4 when it comes to dementia. Interestingly, APOE4 isn't just bad, and there are evolutionary reasons why APOE4 still hangs about. So when you look at the frequency of the APOE allele in different regions of the world, the more red the color, the higher the frequency of the APOE4 allele. And as you can see, in northern latitudes and in sub-Saharan Africa, the frequency of the APOE4 allele is particularly high. And the reason for that is thought to be that actually in areas of the world with low sunlight, such as high latitudes, and with high rates of infectious diseases, actually being APOE4 offers an evolutionary advantage. So maybe in Western world and on Western lifestyle, APOE is a disadvantage, but in many areas of the world, there are still good evolutionary reasons why APOE4 offers health advantages. So then now I'd like to go on for the last 15 or so minutes of my talk, talking about how APOE4 influences response to dietary change. And this is a very nice overview, which was published a few years ago now, which looks at how APOE4 affects the response to different lifestyle factors, things like physical activity, PUFA intake, saturated fat intake, alcohol intake, and smoking. And the reference bar is always the non-E4 genotype and the optimal level of that particular behavior. So when we look at physical activity in APOE4 carriers, you can see that compared to the reference group, APOE4s are much more susceptible to a deleterious behavior. So, for example, in non-carriers, being sedentary increases your risk of dementia by 80%. But you can see if you're an APOE4 carrier, you're much more susceptible to the effects of being physically inactive. Likewise, when we look at things like saturated fat intake, if you're a non-E4 carrier, you are susceptible to saturated fat, but not to the same degree as if you're an APOE4 carrier. And we'll go on and expand on some of these in the coming slides. This is a very nice study where a Finnish group recruited people prospectively on the basis of APOE genotype. They recruited E3E3, E3E4 or E4E4 and looked at the responsiveness to the National Cholesterol Education Programme diet, which is low total fat, low saturated fat and low cholesterol. And as you can see here, the responsiveness of plasma lipids to this diet was very much determined by APOE genotype status, with the E4E4 group being particularly responsive. And this was confirmed in a more recent UK analysis, which was led by Julie Lovegrove and team from the University of Reading, called the RISK study, where they took people with a saturated fat intake on average of about 17% and put them onto one of 4 diets, varying in the levels of monounsaturated fat or glycemic index. And the dark bars here are E4 carriers. The grey bars are E3, E3, and the lighter bars are E2 individuals. And as you can see, particularly when it comes to the low fat, low glycemic index diet, and you look at total cholesterol, APOE4 carriers again are the most responsive group to dietary fat manipulation. And coming back here to our very variable response we saw in the triglyceride levels in response to triglycerides, we went back and looked at how this triglyceride response varied. So this is the change in triglyceride you see in the group as a whole, with this bar here representing the 11% decrease you saw in the group as a whole. But if you look across the piece, you can see that males are more responsive to the triglyceride lowering effects of fish oils compared to females. And again, being APOE4, seems to be a particularly responsive subgroup. Now, one thing I'd like to mention quickly is this is our most recent data, which looks at things called oxylipins. Now, this is a very busy slide, but when you look at omega-3 fatty acids, EPA and DHA, they're metabolized on to a whole host of bioactive compounds called oxylipins, and they can be epoxy, dihydroxy, or hydroxy. And this is something Rashasala in our group has been looking at over the last couple of years. And this was done in collaboration with Philip Calder and his team in the University of Southampton. And basically your ability to produce oxylipins derived from EPA and DHA would be an advantage. And what we did is we used data from a very nice study design of Philip Calder and his team, which was a 12-month intervention where they fed people fish oils for 12 months, equivalent to 1, 2, or 4 portions of fish per week. And they took blood samples at regular time points over the course of the year. And they also extracted lipids from a whole host of lipid fractions, which is here at the bottom of the slide. And what we've done is we've actually taken the plasma fatty acid, in particular the plasma phosphatidylcholine fatty acid profile. And we've also looked at the oxylipin profile in plasma to look and see if this is modulated by APOE genotype. And what we found is, now this is a tiny bit complicated, but this looks at, on the top slide here, you have EPA and EPA-derived derivatives. On the second panel, you've got DHA-derived derivatives. And on the bottom panel, you've got arachidonic acid-derived derivatives. And it looks at the response at baseline, 3 months, and 12 months. And this is all the fish oil interventions combined. And what you can see is the fatty acid responses over 12 months aren't influenced whether you're APOE3 or APOE4, but your ability to produce oxylipins is very much influenced by your APOE genotype, in that people who are APOE4 are much more able to produce these beneficial oxylipins relative to people who are APOE3, which is a really nice finding, and it could also contribute to the fact that it seems that people who are E4 derive greater benefits from omega-3 supplementation. And I'll just draw your attention to the top panel here, which is the conclusions of Hossein Yassin and his group, who are very active in this area, which is around APOE genotype and response to DHA when it comes to cognitive function, that actually APOE4 seem to be a particular group who have problems of taking DHA up into the brain, but would particularly benefit from increased DHA levels. So in conclusion, APOE4 are a large population subgroup who seem to particularly benefit from lifestyle interventions, and particularly higher EPA and DHA levels in APOE4 are recommended. Now, just a couple of final thoughts in the last couple of minutes. Which is a point Luisa brought up, that when we're talking about personalization of nutrition recommendation, of course, it's not just about genotype. When we're thinking about personalizing dietary recommendations, it should also be based around an individual's physiological profile, their habitual diet, but also things like people's food preference, their cultural considerations, food availability, environmental considerations, etc. What is the evidence, and I'll just move on to this slide here. What is the evidence that personalization actually works, and how do we move to a more personalized approach? And I'm sure this is something that Alfredo and Ahmed will really tease out in their presentations. But I thought I'd draw your attention to this very nice review, which was published just a couple of months ago, which was led by Catherine Livingston and John Mathers in Newcastle. And what it did is it tried to tease out is personalized nutrition associated with greater adherence to dietary change. And it summarized data from 11 individual studies which involved between 57 and 1,048 participants, 1,488 participants. The duration of intervention was 1 to 12 months. And the conclusion they came to was that personalization was indeed associated with greater adherence to dietary change or dietary recommendations, but there wasn't enough evidence yet to tease out whether personalization based on genotype versus based on physiological variables versus based on habitual diet is likely to be more efficacious. But the bottom line was personalization did seem to be associated with greater dietary change based on what we know so far. And of course, the great advantage of genotype over other things is that you're born with your genotype. So you can identify and target populations before they have a risky phenotype or before people develop bad habits. And also when you base it on on genotype, it's very specific and more likely to be effective. So this is my final slide. And just to say some final thoughts, that 50% of the maintenance of health and risk of disease is due to our genotype. APOE has certainly emerged as a really important genotype, which I think going forward can really help us target certain individuals who would particularly benefit from dietary fat manipulation. Just to remember that common gene variants are predictive and certainly not deterministic. And always this really good saying that genetic loads the gun and environment pulls the trigger. And that going forward, even though we're probably at the early stages, that genotype is certainly likely to make quite a significant contribution to our endeavours to more personalised approaches to disease risk prediction and to the type of guidance we give to people to control their risk of disease. And it just leaves me to thank lots of people in Norwich at UEA, particularly Anna Luce, Matthew, Rasha and David, and my collaborators in various centres in the UK, and particularly also Gerald Rimbach and his team in Germany. And I thank you for your attention.
Okay. Thank you. Thank you very much, Anne-Marie, for this very nice presentation and for sticking to the time. We have time for some immediate questions. And in fact, there is one question for you, Anne-Marie. It says from an anonymous attendant, he or she says, "Hi, Professor Anne-Marie, thanks for your input. Could you please explain again on the differences between APOE 2, 3, 4 genes?"
Yeah, so it actually affects the structure of the protein. So the protein itself is 299 amino acids. And the genotype affects the amino acid at position 112 and 158 in the protein. So there's 2 amino acid changes in the protein, which affects the configuration of the protein and its stability, and it also affects how the protein is produced and handled within the cell.
Actually, I also wanted to ask you, Anne-Marie, about that. Would you mind sharing your slide, your very first slide showing the SNPs?
Yeah,
because
yeah, I might have to take a second to bring it up. Yeah, so it affects, the SNPs affect the amino acid. So this is the normal protein, which is APOE3, which has cysteine at position 112 and arginine at position 158 in the protein. If you're APOE4, you have arginine at both positions. And if you're APOE2, you have cysteine at both positions in the protein. So those 2 amino acid changes really affect the stability of the protein and the functionality of the protein.
Yeah, and this brings Anne-Marie to my other question, because APOE is circulating lipoproteins, right?
That's right.
Because it is amazing how it can be implicated in the deposition of this protein related to Alzheimer's and so on. So the mechanistic link is,
yeah.
or do you?
Yeah, it's a really interesting question. I mean, most of the protein is produced by the liver, but actually there's quite a lot of protein as well produced by the glial cells in the brain. And actually it's thought that it's the locally produced protein that affects brain function rather than the APOE produced by the liver.
Okay.
And actually, even though there's lots of different apolipoproteins in the circulation, APOE is about the only lipid transporter in the brain, apart from apolipoprotein J. So I think its effect on lipid transport in the brain is likely to be far more consequential because it's about the only protein, it's about the only apolipoprotein produced to any significant amounts in the brain.
Okay. I see. There is another quick question, and since we have time, I think we can do it now. It says, hi, Professor Anne, besides Japanese, a well-known population for longevity, do you have a chance to look at in other Asian populations, such as in Southeast Asians or South Asians?
Yeah.
So
I mean, that's a really good question, actually. I don't think there's been any, to the best of my knowledge, any particular focus on the penetrance of genotype in South Asian populations, but that would be a really interesting thing to look at, but not to the best of my knowledge at the moment.
Okay, so Anne-Marie, thank you very much. If I understood well, the APOE4 genotype is at risk of many things, shorter life, dementia, and so on, but it responds better to dietary intervention with omega-3 fatty acids, right?
Better to, yeah.
Is that correct?
That's absolutely correct. So,
okay.
so they're much more responsive to dietary change than the other genotypes, which has kind of real preventative opportunities for us.
Yeah, sure. Thank you very much. Thank you. So there will be time for other questions during the general questions and answers session at the end of after the presentations. So it is my pleasure to introduce now our next speaker, Professor Alfredo Martínez. He's Professor of Food Science and Nutrition. He has been President of the International Society of Nutrigenetics and Nutrigenomics, and is currently the President of the International Union of Nutritional Science. He has many and broad research interests. His main research interests are in the nutritional control of metabolism, the nutritional utilization of functional foods, the evaluation of nutritional status in different populations, nutrition and immunity, and cell, animal, and human intervention and epidemiological studies in obesity, with a focus on obesity and personalized nutrition, including precision nutritional omics, which will be the subject of his presentation. Professor Martínez is adscribed at the University of Navarra, FIBER-OBN, and in their Research Institute on Food and Health Sciences in Madrid, Spain. So Alfredo, thank you very much. I apologize for this terrible moment. Okay. So the floor is yours.
Okay, thank you very much for this kind presentation. And of course, I want to mention that this activity carried out by MDPI editorial for the journal Nutrients has no conflict of interest, and in particular also is given in the name of the IONS Task Force about Precision Nutrition. Thank you very much for the invitation. When we look to the omics for precision nutrition, often we are considering individualized nutrition, but indeed precision has some aspect related with personalization, with individual situation, but also try to combine and to integrate as much factor, as much information as possible. Interestingly, the concept of precision medicine and nutrition means that is not as new as could be thought by many of us, because Hippocrates already mentioned that your food is the basis of your health. The very well-known physician Galen defined personal attitudes and unique response to food. Later, Claude Bernard and others, they say that no disease but patience. Furthermore, Mendel considered that individualized traits transmission among generations as the father of genetics, and in the 20th century and the early 21st century, you consider that they have been important approach in order to define and to apply precision medicine and nutrition. Indeed, precision nutrition involves the integrated metabolomics for personalization. Therefore, you can see that the individual's metabolism need to consider nutrition, physical activity, the phenotype, personality. But overall, we have an impact of environment, but also an impact of the background related with genetics and the microbiome. Therefore, precision nutrition involves the interpretation and the integration of personal information, but also information that is related with the environment. Deepening this concept, the phenotype of traits that a person figure out depends on the genotype, as mentioned by the previous speaker, Professor Minihane. Therefore, precision nutrition takes into account the genotype, the microbiota as internal and endogenous factor, but also takes into account environment. In other words, the gene expression as shown by the phenotype depend on endogenous factor and the interaction with exogenous factors. Therefore, in these introductory words, precision nutrition benefit a lot from omics. As you know, the genome, through genomics, the mRNA determination of transcriptome analysis through transcriptomics, microRNA, the proteome, lipidome, depends on proteomics, the metabolic analysis depend on metabolomics, and recently, of course, the microbiota is the source for metagenomics. Therefore, omics or overall techniques is an important support for precision nutrition. These omics tools based on genomics, transcriptomics, proteomics, lipidomics, metabolomics, metagenomics give content and help to apply precision nutrition ideas based on these omic tools. My first part will be considering the role of genetics in personalization. As all of you know, genetic influence dietary choice and food intake, but also genetics influence processes like digestion, absorption, transport, metabolism, and also excretion. The real question is how to interpret this different genetic background, the genetic makeup, and the interaction with nutrients. Of course, we all agree that there are possible interaction of diet and nutritional component with genetic variability to affect health and disease risk. Therefore, I can show here an example where a given genotype by carrying this allele have an impact on waste-to-heat ratio, but also in the respiratory quotient. There is an idea that mutation in a given gene can affect phenotypical expression. The example that I have shown at the beginning have developed the concept of the GWAS, Genome-Wide Association Studies, and in this case, we have information that early onset extreme obesity in French and German groups have been related with this message, but also the genotype influence the HDL cholesterol level, or there are different loci that contribute to polygenic dyslipemia. just depending in this concept, different studies has related the effect size of genetic variants related with BMI. You can see, for example, that the carriers of alleles related to FTO can have about 1 or 2 kilos or 2 extra kilos of weight just by being carrier of this allele. Difficulties comes when the situation, you see, this is the metabolic genome, that there are many genes related with metabolic homeostasis. In the case of obesity, you can see in the bottom, FTO, MC4R, TMEM, UCPs, leptin, etc. There are genes that are known to regulate energy and nutrient metabolism. Therefore, two concepts has come to the floor. One is Nutrigenomics and the other one is Nutrigenetics, that are based on omics science, are related to define the possibilities of this nutrient and gene interaction. As you know, Nutrigenetics investigate the genotype influences on the response related with nutrition and personalized metabolism. A clear example is phenylketonuria that inform us and condition and jeopardize the situation if you are eating too much amino acid as phenylalanine. The other side of the coin is Nutrigenomics. These are the dietary effects on gene expression. Therefore, an example is how sugar or sweet consumption increase the expression of insulin in just a few minutes. Therefore, the interaction of the genotype and gene expression should be considered when we are talking about precision nutrition with an omics perspective. An example, how nutrition influences gene expression and can, at the end, influence phenotype. You see, this is a classical experiment concerning the European project Nutrigenomics. As you can see, the expression of different lipid metabolism genes is affected by the content of the diet and also within an energy restriction protocol. Therefore, we can see that omics technologies are able to determine transcriptomics examples of expression of genes related with lipid metabolism. As mentioned, also the genotype can influence nutrition, and there are some examples of changes in gene function, gene expression, with impact on the phenotype or nutritional status. A few examples to remember omics possibilities in this area. As you can see here, individuals that eat more than 49 of energy from carbohydrate that carry out the mutation, this mutation, are able to show higher BMI levels. You see the column in green. Therefore, there is a clear interaction of carbohydrate intake with this mutation. As the concept has evolved, now we can use genetic reach tool to check obesity predisposition and interaction with macronutrient intake. You see, it's clear that total energy increase is associated with increases in the percentage of body fat, but those in a low GPS, a low genetic predisposition score, with less than 7 risk allele, you can see here that they show lower amount of body fat at the same level of total energy intake. Another example is that, as you see, the genetic predisposition score has an effect, an impact condition the percentage of body fat. You can see here the polyunsaturated fatty acids or fiber intake or vegetable protein have differential effects depending on the genetic score. In some cases have different direction or have different slopes. Therefore, there are examples where classical example with genetics has an impact on the situation of the phenotype related to the percentage of body fat. As mentioned, the situation is complex because there are many hundreds of interactions. I am going to mention some of them related with MTHFR receptor, with an interaction on BCs, the gene PMC related with appetite, insulin resistant related with gene is affected by the intake of carbohydrate, and you have APOE5, PPA gamma, beta 2 adrenergic receptor, the melanocortin 4 receptor, and of course FTO. There are many interactions that are related with this information. Genes related with nutrients or nutritional patterns and have an impact on the phenotype and an impact on health. In other words, omics can have a look, help this information. But as I mentioned, going to practical terms, precision nutrition and practical nutrition, you can see here that personalized nutrition can benefit from information, from omics information. You can see here that the people that are carrier of the C allele have a resistance to weight loss, or in other case, the genotype related to yellow, red, or green colors show differences in weight regain. In some cases, subjects with this genotype regain easier, other remains stable, and the other eventually gain weight if they are carrier of this genotype. Perhaps the more clear example I can show here is related with the Food for Me project, where subjects that are carrier of the risk variant of the FTO can benefit from the information related with BMI, related with waist circumference, can add information related to physical activity, and also with glucose or cholesterol. Depending on genetic information, the genetic makeup, but also phenotypical information and lifestyle, we can use this omic technology, this nutriomic technology, to prescribe more personalized diet with more precision. Again, you can see here that weight loss depends on many genes, and of course, at this moment, it's difficult to recognize and apply all the interaction that have been identified but need to be integrated for a clearer and more practical practice. In any case, I am going to show you an example. If we have information about sex, age, initial body weight, important SNPs related with obesity and weight loss, the energy intake, and of course physical activity, we can just promote one diet or other based on preferences of the subject, based on the exchanges lists. You can see here that person 1 or 2, depending on the genotype, phenotype, and lifestyle, can be just recommended diet 1 or diet 2. Diet 1 is related with protein, moderate protein intake, and diet 2 is a low lipid, low fat diet. Once I have shown some nutrigenomic application and value for this area, I am going to move to a second omic application in this area, is epigenomics. Epigenetics literally means in addition to change in genetic sequence. As you can see there is a situation where DNA methylation and histone modification are related and can be applied for precision nutrition. As all of you are aware, DNA methylation, histone modification, chromatin packaging, DNA packaging, and of course, enzyme modulation through microRNA are phenomena, are processes with epigenetic possibilities to be modified and affected by diet. I am going to show one example. This is a group of subjects that were submitted, that were offered a 8 weeks hypocaloric diet. Some of them responded well and were good responder, and the other were low responder. What happened with the omics interpretation of these epigenetic changes? You can see here that there are a group of CpG that were hypermethylated, and some of them they were hypomethylated before and after the diet. Of course, you see some of them they are important genes that you all know important in energy regulation. Therefore, there are epigenetic changes as a consequence of energy restriction. But also in this same experiment, there is differences in low and high responder concerning epigenetic biomarker of weight loss. You see interleukins and other sirtuin caspase that show differences between subjects that were well responder or high responder as compared with subjects that lose a small amount of weight. But there is more application of omics in obesity and metabolism investigations and application. You can see here that sequencing of microRNA in peripheral blood mononuclear cells is able to identify potential weight loss subject. This is an example. In the app, you see that responder have lower levels of 2 microRNA. However, in the bottom of this slide, you can see that microRNA are marker of subject that can be good or bad responder. In other words, here we have the concept that microRNA could be markers or biomarkers of future weight loss when following a low energy calorie diet. The third example that I'm going to show you about omics application rely on metabolomics. This is an example that the metabolomic profile in the urine is able to follow the intake or the adherence to a diet, but also to be related with biological markers. As you can see here, blood biomarkers of obesity and comorbidities depend on genetics. We have talked about that. Depend of lipid markers, depend of age, depend of race, depend sex, and also depend on lifestyle like smoking and physical activity. Current situation of metabolomics is able to have more information about the glycemic profile, lipid profile, vitamin D, and also inflammatory, oxidative, and neuroendocrine biomarkers. The example that I'm going to bring to you as a possibility of omics technologies to be applied in nutriomics are how can measure metabolite in urine through a HPLC TOF model. As you can see here, either with the negative mode or the positive mode, we are able to follow the adherence to the diet. There are differences, the light blue as compared with red or dark blue, once they have followed the control group or they have followed the cocoa diet. Therefore, metabolomics can follow the attrition, the adherence, but also in this case was able to relate a BDI marker related with, with depression and a metabolite like this hydrovanillic acid. Therefore, we are able in this example that we can relate metabolomics for nutritional application, in this case related with cognitive and depression activities. The fourth example that I want to show you about nutrition and omics related to gut microbiota. As you know, some scientists, not all, there is no consensus, consider that good microbiota is a new organ within an organism. Different levels of this phylum to a species level are known to regulate host function, immunity, nutrient production, or energy harvesting. You can remember that precision nutrition can rely on gut microbiota because it can inform about metabolic functions, can inform about structural and protective functions, and immune function. In other words, omics related to obesity and gut microbiota can inform about diversity, about dominance of specific microorganism, also metabolic production of short-chain fatty acid, intermucosal barrier, not over inflammation. You can see that this omics is able to differentiate obese and non-obese. This is a phylogenetic diversity depending on microbiota in lean and obese subjects, but also fecal microbiota is differential between lean and obese mice depending on the APOE genotype. This has shown some application. This is a classical experiment where, as you can see there, a calorie-restricted diet and an increased physical activity, there is a differences in subject depending on the high weight loss group as compared with the low weight loss group. In some cases, there are changes in some species, but not only that, there is a correlation of different material group with weight loss, therefore can be used for prediction. I am going to start with the summary of this presentation. Nutrigenomics, in other words, omics technology used or applied in nutrition relies on the knowledge of gene and environmental inputs, metabolomics, and microbiota interaction. Therefore, omics can benefit information related with genomics, metabolomics, and metagenomics that if we adequately integrate, we can have a situation where we can interpret, we can just define individual response, precision outcomes in the subject, and therefore we can prescribe better advice to these subjects. Therefore, we need to have information of genetics, we have information on genomics, but not only that. In nutrition, also it's very important to have information about nutrition, taking into account that we need to consider different food, different amount of food that also can benefit from omic and big data information. Also, we need to consider information related with physical activity. Also, physical activity can benefit from omics related with the specific genes, but also from metabolomic information related with physical activity. Genetic and genomics is another tool that can be used with an omics perspective. And as mentioned before, epigenomics is important, but not will explain everything. To summarize this group of ideas, genomics, metabolomics, epigenomics. Also, we need to take into account metagenomics. In other words, the information about gut microbiota can be useful for precision nutrition. Diet and specific components like polyphenols, probiotics, symbiotics, antibiotics or antimicrobials can be important to interpret and to use precision nutrition and to manipulate gut microbiota with effect on health maintenance or disease prevention. Just to take into account that dietary, phenotypical, and metabolomical data needs to be considered in a holistic perspective and strategy, because it's very important in nutrition to have information about food consumption, about the impact on health, therefore in cholesterol, triglycerides, glucose, insulin, and also more specific metabolite, as you know, for example, now liver disease can be very well characterized based on lipid species characterization through omics perspective. Therefore, personalization for weight and health maintenance depend on diet, phenotype, and genetics. My final is that precision nutrition is not far away, because we have information about the genome, we have information about gene expression that, as I mentioned, depend not only on DNA, but also on nutrition, physical activity, and environmental interaction. But of course, yeah, nowadays, we have real application, because omics can be used for define specific nutrient requirements and recommendation. Omics technology are able to help us in predisposition and nutrition-related disease, and of course, the open space appear for prescription of diet in order to have roles in prophylaxis or roles in therapies. Precision nutrition-based genomics, therefore, clarify the idea that there is no unique optimal diet, but there is a range of adequate diet depending on genetics, biological, and cultural variation, where omics are important. Therefore, omics is able to provide genomic information, to provide information about allergies, intolerance, epigenetic marks, and also the characteristics of a personalized diet. My last words is to invite all of you to the next IONS-ICN meeting that, given the situation of pandemic, the COVID pandemic, has moved from September of this year to December 22. You are all invited and ready to provide answer or comments either from you or addressed by the chair, Professor Luisa Bonet, if you have explained this in the chat. Thank you very much, and ready to comment your ideas where omics technologies can be used in precision nutrition. Thank you very much, and ready for answering. Thank you.
Okay. Thank you very much, Alfredo, for this nice presentation and very broad and complete presentation. So there are a few questions, and since we have some time now, I think I can translate them to you now. Claire Hebert says, "Thank you for this fascinating presentation, Dr. Martinez. She has two questions. One is, given that obesity itself can change in expression, is there a way to tell whether a metabolic condition has changed gene expression versus whether an epigenetic change has caused the metabolic condition?"
Thank you very much for your very interesting question. Indeed, there are examples, I chose some, but there are many others, that genetics can influence the epigenome. And of course, there are examples where obese have differential methylation patterns as compared with lean. And also, there is example where epigenetic can predict weight loss. And more interestingly, as you mentioned, there is a situation where epigenetics can be changed depending on the diet. It's susceptible, and there is an open window for future actions in this field, how to change epigenetics in order to benefit the response to energy restriction.
Okay, thank you. The second question from Claire Hibert is, do you know the mechanism whereby genes make some people more susceptible to weight loss or weight gain? So what are
you can find some reviews where there is a list. For example, there is one review about this point, and I show a part of this in my presentation, that there are a susceptibility of different genes that can help to predict and of course prescribe different diet. Currently, there is about 100 genes related with weight loss, with appetite, with lipid metabolism, adipogenesis, etc., that can help to understand the obesity of a person. The real challenge, as you are aware, is to identify, characterize, and to feature the interactions between different alleles and the outcomes they produce or they generate. Therefore, there are about 100 genes that are related with weight loss or obesity, but the difficulties come from the interpretation of the interaction. Thank you.
Okay. There is another question from an attendant, Ruhana Mediti. The question is, he says, insightful information. Can you please explain the difference between precision nutrition and personalized nutrition? Is there any difference between precision nutrition and personalized nutrition?
Thank you. I am going to give my personal view, my own view, but it means that not everyone is in agreement with this. For me, personalized or individualized nutrition refers to the endogenous factor one person carries. For example, genetics and microbiota. They are personal items. However, there are another personal factor that can be external affected or can be just influenced by different situations like smoking, like physical activity, like living in a high altitude, etc. Therefore, precision nutrition involves the consideration of as much as possible external factor that have an impact on health, on the phenotype. But also precision nutrition have another perspective, is the interactions of this endogenous factor like genetics and microbiota with external. Therefore, precision is a more elaborated concept, taking into account personalized factor, endogenous factor with exogenous environmental factor with impact on health. Precision nutrition tries to account the interaction between individualized matters with those factors that are able to be selected or chosen by the subject of the individual. Thank you. Hope. In any case, if you don't agree with this, I can go further, because I understand that in some cases, precision and personalized means the same. For me, it's not the same.
Yeah, actually, I think your answer already answers the last question at the moment, which is from an anonymous attendant. He says, nice presentation, Professor Martinez. I have a question here. Do we need to consider the psychological health and sleep quality of an individual in precision nutrition? Thank you.
Of course,
Of course.
behavioral matters, behavioral issues are very important. Some of them are related with the person because perhaps they have a component, a genetic component, but others are personalized because they have an influence from external cues. Therefore, of course, psychological, behavioral, and mood, and depression, and quality of life should be taken into account in precision nutrition. And of course, we need to be objective. Therefore, we need to create a score that taking into account behavioral and psychological signals.
Vale. There is another question for you, but from Tassier Chinedu. He says, thank you, Professor, for the presentation. Please, in what ways can personalized nutrition reduce the chance of becoming diabetes in persons who have a strong positive family history of type 2 diabetes mellitus?
In this case that you are mentioning, thank you for the question. There one aspect, of course, is if you have genetic predisposition in the family, you can check a number of SNPs that inform you about predisposition. But perhaps also this question can be complemented by Professor Minihane. But coming back, the genome informs you about your susceptibility or your predisposition, but not about the penetrance. You need to take into account other factors that can influence diabetes, and you can take care. Perhaps a very drastic example comes from the Jolie, for this actress, that knowing that her mother and her sister have gynecological cancers, they remove the ovaries and part of the mammary glands in order to avoid this very high risk she has. Therefore, Angelica Jolie take the dramatic way. Other ways is if you have a predisposition, you can just prevent, just taking healthy measures that you know that are related with diabetic, hypertension, or any disease impact onset. Therefore, there are two approaches: just measure or evaluate the genome and also to take healthy measures. Thank you.
Okay. There is a comment also from Sruhana Mediti, going back to the concept of precision nutrition. So can we say chrononutrition also plays a major role in personalized nutrition? Chrononutrition, personalized nutrition.
Of course, of course. This is very important because personalized nutrition, you know that people, they prefer wake up early, and others they prefer to go to bed late. This is, some of them is a condition of genetics, but also there is an attitude of familial influence or social influence or labor influence. Therefore, chrononutrition relies on personalized nutrition, of course, both in genetics, but also in environmental aspect. I don't know how do you say in English. Perhaps, you know, there are some birds, in Spanish we say alondra, that wake up early, and there are birds that remain all the night awake, like búhos. Therefore, there's genetics, but also there are some conditions if you live in a country like Sweden, or if you live in a country in the Ecuador. There are differences, but some of them they are related with genetics, and the other are related with the environment. Thank you.
Okay, thank you. Thank you very much, Alfredo, for your presentation and your answers. Now, I remind you that there will be a joint question and answer session after our next presentation, which is given by Professor Ahmed El Soemi, who is now on. Hi, Ahmed, are you ready?
Hi, Luisa, yes, ready to go.
Okay, then I will make your presentation. I will introduce you first. So our last speaker, we start 5 minutes before the scheduled time. Is that okay, or you prefer? I think we can start now and continue.
Sure, yeah.
So Professor Ahmed El Soemi is professor and Canada researcher in Nutrigenomics. His research in Nutrigenomics and Nutrigenetics aims to identify biomarkers of dietary exposure and elucidate the genetic basis for variability in nutrient response and dietary preferences. His research group has also been studying the effects of disclosing genetic information on eating behaviors and exploring attitudes and perceptions of genetic testing. Other research projects of his group focus on micronutrient genomics, genetics of caffeine response, and genetic determinants of eating habits, as well as athletic performance. And his presentation today is entitled Genetic Testing for Personalized Nutrition. Actually, we have been now having a discussion on whether there is a difference between personalized nutrition and precision nutrition. Professor Ahmed El Soemi is in Canada, and I remind you that it's early in the morning now in Canada, so that's why he joined the seminar right now. So that's why I'm saying this about this discussion. Okay, so Ahmed, the floor is yours. Thank you.
Great, thank you very much, Luisa. And I guess just in terms of Alfredo's comment about early birds and night owls, I'm definitely a night owl. So not an early riser, but hello and good morning from Canada to everyone. I just want to start by thanking Charlotte and the team from Nutrients for organizing this webinar series, and Dr. María Luisa Bonet for chairing this, and my fellow speakers. I wanted to share with you my perspective on this topic of genetic testing for personalized nutrition, but also touch on some of the other elements that the other speakers also refer to in terms of aspects of personalizing nutrition. So just by way of disclosure, I am the founder and I do hold shares in a company called Nutrigenomics with an X, which provides genetic testing for personalized nutrition only for healthcare professionals. So this is not a direct-to-consumer company. Now, we've already heard, and Anne-Marie gave a great overview of genetic diversity and the different kinds of genetic variants. And as we know, of course, there are millions of genetic polymorphisms throughout the human genome, primarily single nucleotide polymorphisms. These are the most abundant and perhaps the best well-characterized form of genetic variation. And obviously, if we have particular polymorphisms, they can impact the way we look and function. And as you can imagine, if polymorphisms can affect our external features, you can imagine they also affect our internal workings, genes that affect the way we absorb, metabolize, and eliminate virtually everything that we ingest. And so the interest in my lab has been trying to understand how these common genetic variants modify our response to various dietary factors. So the science of nutritional genomics or nutrigenomics, one aspect of that I would say is using genetic testing to determine and understand why some individuals respond differently from others to the same foods, beverages, and supplements that they consume. Again, both Anne-Marie and Alfredo gave some great examples of how this occurs. From a research perspective, we can really think of two sides of a coin. On the one hand, illustrated by the upper arrow, we can think of how diet can affect genes. Nutrients can upregulate or downregulate the expression of various genes. Nutrients can also alter or cause epigenetic modifications. What's illustrated by the bottom arrow, and really the focus of our research efforts, is understanding how variations in genes affect the way we respond to diet. So again, genes that code for proteins that affect all aspects of metabolism, absorption, digestion, distribution, and elimination of everything that we ingest. So one question, though, that some I guess more traditional nutritional scientists wonder is, why bother with genetics? Why do we consider that it's absolutely essential to take into account genetic differences when studying nutrition? And I think if you trace the path from virtually any nutritional factor to any health outcome, if there have been enough observational studies, we tend to see this heterogeneity of response. And again, that was illustrated nicely, I think, in one of the waterfall plots that Anne-Marie showed, where fish oils actually increase the levels of triglycerides in some individuals. They have no effect in others, and yet decrease it in yet other individuals. A lot of different reasons for these inconsistencies or heterogeneic responses, but an important aspect, I think, is the genetics of the individual that's being studied. And I've tried to illustrate that in a black box, where if we can predict a person's response based on their genotype, then perhaps we might be able to tailor the recommendations that we give more specifically to that individual. So clearly, one size does not fit all. And it's a concept that we've known for a really long time. In fact, one of the earliest references comes from the 1st century BC, where Lucretius noted that one man's food is another man's poison. What's good for one person might actually be harmful for someone else. So we've come a long way in the field of nutritional genomics and molecular genetics, where we can now pinpoint specific regions of the genome that can help us predict if a particular nutritional intervention might be beneficial or harmful for a given individual. And this is clearly, paving the way towards personalized and precision nutrition. So where or how can genetics impact our response? Well, first of all, they can influence our sensory perceptions. Genes code for taste receptors on the surface of the tongue, as well as olfactory receptors in the nasal cavity that influence our likes and dislikes of various foods. And these actually influence the types of foods that we choose to consume. Some people, for example, have a great sensitivity to the bitter taste of certain cruciferous vegetables, and they choose not to consume them because of that genetic aversion to those particular nutrients or those foods. Genes can also influence our appetite and sense of satiety. So some people need more of a particular macronutrient to get the same satiating effect. And of course, these are factors that influence what and how much we consume once those foods are ingested. As I mentioned earlier, all kinds of genes that code for various proteins, transporters, enzymes, receptors, binding proteins, they all influence how much of a nutrient can circulate in the system, and ultimately what reaches the target cell of interest to exert a particular biological effect. So a number of years ago, back in 2007, I was at I think it was the first Nutrigenomics, the Bruce Ames Nutrigenomics conference at UC Davis. And the headline in the newspaper the next day was "Genetics may define diets of the future." And as though this concept was something that was very futuristic, that people would actually start eating according to their genetics. But it's actually not that futuristic. It's something that we have today. There are many products on the supermarket shelves that are actually developed for people with particular genetic variants. An obvious example, of course, are gluten-free foods, which essentially are meant for those with celiac disease, who have specific genetic variants that result in gluten intolerance in the HLA gene region. And likewise, lactose-free products. So individuals that have a particular gene or a variant in the LCT gene that makes them unable to produce enough of the lactase enzyme in early childhood and adulthood, they're unable to metabolize lactose and they develop gastrointestinal discomfort. So here are examples of products that are developed for individuals based on their genetics. Now, not everyone who has lactose intolerance knows their genotype. They just know it by trial and error, because lactose intolerance is one of those conditions where you can probably predict if you have it based on your response to say, consuming a glass of milk. So we were interested in understanding the physiological consequences of lactose intolerance. And just a couple of years ago in the Journal of Nutrition, we reported that individuals who have the genetic variant that predicts lactose intolerance, and that's illustrated by the white bars. So those who have the CC genotype have full lactose intolerance. Individuals who are heterozygous, those illustrated by the gray bars, have intermediate phenotype, and those with the black bars have no lactose intolerance. And you can see both in the total population as well as women and men separately, that individuals who have lactose intolerance, or at least the genotype that predicts it, have significantly lower blood levels of 25-hydroxyvitamin D. And we know, of course, that vitamin D has been linked to all kinds of health conditions. And you can see here that individuals who have lactose intolerance, which we know they self-regulate how much dairy they consume, and given that dairy is an excellent source of vitamin D, they're clearly not getting adequate amounts of vitamin D in their diet or from any other source. So even though they have lactose intolerance and they may or may not be fully conscious of the fact that they're limiting their dairy intake, they're clearly not compensating for this by getting adequate amounts of vitamin D from other sources. So the message really is that those who have this genotype that influences an eating behavior, in other words, avoidance of lactose-containing dairy products, these individuals are not compensating and getting vitamin D from other sources. And I should mention that we use this approach called Mendelian randomization, where you effectively use a genotype to predict an exposure. And here it's the LCT gene that predicts exposure to lactose-containing dairy products. So it was the kind of thing that got a bit of media attention. And again, while it might seem obvious, those who have lactose intolerance are limiting their dairy intake. It's not so obvious that they're actually not compensating by getting enough vitamin D from other sources. So that, I think, is an important public health message. It's almost like, you don't need to have your genetic data to understand that if you are limiting your lactose or dairy consumption, you need to ensure that you're getting adequate amounts so that you can optimize your blood levels of 25-hydroxy-D, which is, as all you know, of course, has been linked to all kinds of health conditions. More recently, we also used this approach of Mendelian randomization to study the links between iron metabolism and premenstrual symptoms. So this is work that was done by a current PhD student, Tara Zaytoun, who has been investigating the role of various nutritional factors and genetic modifiers on premenstrual symptoms. This is a condition that affects the quality of life for many women, and it's unfortunately have been understudied for many years. And so we think it's an important aspect of women's health that warrants this kind of research. And what we've shown here is that individuals who have the genotype that predicts iron overload, in other words, at high iron status, they actually have lower likelihood of experiencing some premenstrual symptoms, but not others. So shifting gears a little bit and looking at some of the more common chronic diseases, cardiometabolic disease being the #1 cause of death among US adults. There was this article in JAMA a few years ago that estimated that approximately 50% of cardiometabolic deaths are attributed to a poor diet. What was not clear, of course, is what aspect of the diet is responsible for this, and more importantly, in which individuals. So work that we've done looking at cardiometabolic disease going back over 15 years ago now, which we reported in JAMA, looked at the link between coffee, genetics, and heart disease. And I'd just like to use this to illustrate the concept of where we can use a common single nucleotide polymorphism to predict a person's response to an exposure, and in this case, it's coffee consumption. And what you can see here is on the left-hand side, those who have the APOE genotype, which makes them fast metabolizers. In other words, they get rid of caffeine very efficiently. Moderate consumption is actually protective. So 1 or 2 to 3 cups a day lowers the risk of heart attack. Whereas on the right-hand side, carriers of the C allele, even 2 to 3 cups a day is linked with a significantly higher risk. And those who drink 4 or more cups have almost a 4-fold higher risk of experiencing a heart attack. So this has since been replicated, but looking at other cardiometabolic outcomes. This is a group in Italy that showed a similar phenomenon, but looking at risk of hypertension, which is of course an important risk factor for a heart attack. And while the cup sizes and groupings are a little bit different, you can still see the same type of pattern. On the left-hand side are the fast metabolizers. Coffee seems to be protective, whereas the slow metabolizers, coffee increases the risk. Likewise, again, another replication study showing that with prediabetes, impaired fasting glucose, coffee increased the risk of impaired fasting glucose only in the slow metabolizers. Another group in the United States, again, back just a couple of years ago, demonstrated a similar phenomenon. They looked at 2 different genes that influence caffeine metabolism or response, the adenosine receptor as well as the CYP1A2, and only the CYP1A2 gene influenced caffeine's effects on postprandial glycemia, again, in the same manner. So all these studies demonstrate that if you're a slow metabolizer, coffee has adverse health effects, or caffeine at least, has adverse health effects, whereas if you're a fast metabolizer, you're lucky, you can consume the recommended intake levels. There's actually been other studies that have replicated this as well, looking at systolic and diastolic blood pressure and other outcomes as well, all showing the same thing. And again, when it comes to the research, it's important for us to look at replication because in the field of population genetics, some of the earlier genetic association studies failed to replicate those associations, and which is not really surprising because we know that genes do not act in isolation. They interact with their environment. And again, this is an example showing that a single nucleotide polymorphism that affects the rate of caffeine metabolism can predict a person's response to caffeinated products. Shifting gears again and just giving another example of the benefits and value of using genetics to personalize a person's diet. Weight management, of course, is a a huge industry, an important health area. It's more than a $70 billion industry in the United States alone. And I just want to emphasize that we're not trying to use genetics to predict obesity. You can simply just, if you're a clinician and you see patients, you can just have them stand on a scale or measure their body fat, and you can tell whether or not perhaps they can benefit from losing weight. The question really we want to ask is, how can we use, or can we use, genetics to determine the best dietary approach for weight loss? Now, there's all kinds of fad diets out there, low fat, low carb, high protein, South Beach diet, paleo, Atkins, you name it. And the reason why a lot of these fad diets persist is because they actually work, but in some people. So the question is, can we use genetics to predict which of these types of dietary strategies might be more effective? Most people just do it by trial and error. That can, of course, lead to frustration if a particular diet does not work, and then people just kind of give up and say, oh, you know what, I'm destined to be overweight or obese. It's in my genes and there's nothing I can do. Well, the field is not without controversy, and some people often refer to the Diet Fit study that was published a couple of years ago in JAMA as showing that genetics cannot be used to predict a weight loss diet. And you see some of the headlines, your DNA won't determine the best diet to help you lose weight, and here's why matching your DNA is a waste of time. Well, if you look carefully at the Diet Fit study, you have to look deeply into one of the appendices to find out which polymorphisms they actually looked at. And they decided this way back in 2010, and they selected 3 SNPs, one in the fatty acid binding protein, one in PPAR gamma, and one in the beta adrenergic receptor. And what's really puzzling is none of these SNPs have ever been associated with weight loss, let alone weight loss in response to low fat or low carb. So these SNPs were actually selected because there was a company called Nutrigenetics, which is no longer in business, was marketing a test based on these 3 polymorphisms. But it's really bizarre because these polymorphisms have nothing to do with response to a low fat or low carb diet. The analogy is looking at a gene, the lactase gene, and looking to see if it predicts your response to gluten or to caffeine. It just doesn't make any sense. So it's not surprising that they found that these 3 genetic polymorphisms do not predict response to low fat or low carb diet. But when you look at their waterfall plot, again, similar to the graph that Anne-Marie showed with triglyceride response to fish oils, you can see that for each individual, illustrated by a line on this graph, those who are on the low fat diet on the top, those who are on the low carb diet on the bottom, and weight change. You can clearly see on the left-hand side, a lot of individuals who lost a great amount of weight on either the low fat or the low carb diet. But as you move towards the right, you can see that some individuals on the low fat diet actually gained weight. And likewise, some people on the low carb diet also gained weight. So clearly there was huge variation in response. And I think one of the most important determinants of this variation is a person's genetics. We just don't know what those genetic polymorphisms are yet. So I think when we think of the Diet Fit study, we shouldn't look at it as a failure of genetics. I think we should look at it as an opportunity to identify genes that can predict these differences in response in some of the individuals. Where I think the science has been more robust and really promising comes in the area of a high protein diet. And this is a study that was done by Luke Hughes' group at Harvard, where they conducted a 2-year randomized controlled clinical trial comparing, so this is the pounds lost trial. So they're comparing the effects of a low protein diet to a high protein diet. And as you can see, the amount of body fat is illustrated on the y-axis really it depends on the version of the FTO gene that the person has. So if you're a carrier of the T allele, if you have the TT or TA genotype, there's really no difference in how much body fat you lose, whether you're on a low or high protein diet. But take a look at those on the right-hand side, those who have the AA genotype, which we would call the responders. Those individuals who were on a high protein diet lost a considerably greater amount of body fat, and they maintained it 2 years later. This work has since been replicated. A group in Spain also showed the same thing, that a high protein diet only benefits those who have the FTO gene. This is a slightly different SNP, as you can see by the RS number, but those two SNPs are actually in complete linkage disequilibrium. So they essentially measure the same genetic variation. Excuse me. Another group in the United States also looked at the response of a high protein diet in a short-term weight loss trial after only 4 weeks on a hypocaloric diet that was either low or high in protein. Again, the high protein diet only benefited those who had the risk genotype in the FTO gene. We also replicated this a few years ago. This was work that was done by one of my summer students, Dave Merritt, and this was a cross-sectional study in a multi-ethnic population. And we published this in Genes and Nutrition back in 2018. And you can see here, we looked at waist circumference. And if you remember, those who have the AA genotype, those are the individuals who benefit most from being on a high protein diet. And you can see that here. Those individuals with that genotype who were not on a high protein diet, they had a considerably greater waist circumference, approximately 10 centimeters. Whereas in those who had the T allele, there's no difference between those who are on a low protein diet or a high protein diet. Again, emphasizing the impact of a high protein diet on weight loss is only in those who have the AA genotype. So if you don't have that genotype, you might have heard of a high protein diet. The evidence to date really shows that it's not likely to work if you don't have the AA genotype. Now, it's not really clear what the mechanism is, but we know that FTO does impact food intake, and we know that protein can be satiating, but it has a greater effect in some individuals than others. So it's quite possible that a high protein diet improves satiety to a greater extent in those who have the AA genotype. Now, one question that some individuals have is, well, what happens when you give people their genetic information? And I know that Anne-Marie touched on this with the Food for Me trial. We asked this question a number of years ago, and we wondered if, on the one hand, some people might say, yes, I have the risk genotype. It's going to motivate me to eat a better diet. The flip side could be this, and the concept of genetic determinism or fatalism. Some might argue, I have the gene, there's nothing I can do, pass me the remote and potato chips and leave me alone. Well, this concept really relates more to genetic predictors of disease risk. But when it comes to the modifier genes, if you tell someone you're a slow metabolizer of caffeine, there is actually something they can do. They can limit their caffeine intake. If you tell someone you have the gene for salt-sensitive hypertension, you need to limit your sodium intake. That's something that's very specific and actionable that they can do. We're not just telling someone you have the gene for Alzheimer's disease or heart disease. Good luck figuring out what you need to do. You have to give people the right kind of actionable information. So we decided to put this to the test, and we conducted the first randomized controlled clinical trial in this field that looked at the effects of disclosing genetic information. And this was work that was done by a former PhD student, Diva Nielsen, who's now a professor at McGill in Montreal. And we found that those who got the DNA-based dietary advice actually understood the recommendations. They wanted to learn more, and they were more motivated to change their eating habits. Now, motivation is one thing. Did they in fact change it? we found that yes, indeed they did, and we reported this in PLOS One. And those who got the DNA-based dietary advice actually changed their eating habits, and they maintained it 1 year later, showing greater compliance to the recommendations. And the biggest effect that we saw really was, again, in those who got genetic information for salt-sensitive hypertension. They lowered their sodium intake by almost 300 milligrams, and they maintained it 1 year later. This has since been replicated, this concept of looking at disclosure of genetic information. And this was a review article that was published in Lifestyle Genomics by Justine Horn and her colleagues at the University of Western Ontario. And they concluded that the provision of actionable information is more likely to result in health behavior change. This is distinct from just giving people information on their genetics and leaving them to their own devices to figure out what to do with that information. So it's important to have the genetics coupled with clear and specific actionable information and recommendations. Just last year, Justine published another randomized controlled trial that looked at the effects of disclosing genetic information. And this was a study that they published in the British Medical Journal, Nutrition, Prevention, and Health. And they compared individuals who got the group lifestyle balance, which used to be called the Diabetes Prevention Program, which is really the gold standard for weight management. And they compared those individuals with a group that also got the same diabetes prevention program, but on top of that got a Nutrigenomics test. This was actually the test from the company Nutrigenomics, but it provided information on personalized information on top of the diabetes prevention program. And they found that giving people that genetic report actually resulted in even greater adherence to the dietary recommendations. They also reported, I think in the journal Obesity, greater improvements in body weight and body composition in the group that got genetic information. So how do we balance then the advice that we give to individuals versus the public health recommendations? Some have argued that genetic testing and personalized nutrition is going to revolutionize the field of nutrition. I think that's a little bit of hyping. I think it's going to be more evolutionary than revolutionary. It's really a matter of giving people a little bit more information about themselves and their own unique biochemistries and physiology through genetics to help them and nudge them towards making better lifestyle choices. Some of you may recall that a few years ago, Richard Thaler from Chicago won the Nobel Prize in Economics for the concept of nudge theory, really looking at small ways of making people make slightly different behaviors and choices. And this concept of nudge theory really applies to nutrition as well. And again, I see the role of genetic testing as just another tool that individuals can use or healthcare practitioners can use to help their patients or the clients to nudge them towards making these behavior changes that can ultimately improve their health, because based on their genetics, they're the ones who can benefit the most. Now, as I mentioned earlier, the field of genetic testing is not without controversy, and there are a number of skeptics who have made some claims that I think are just misleading, not to mention false. And I thought it would be important to kind of go through some of those comments. So we've seen a number of articles and blogs talking about how companies are trying to use genetics or your microbiome to give you personalized advice. And here's why this is a waste of your time and money. Some of this actually came out after the Diet Fit study, which was really unfortunate because, as I mentioned earlier, the Diet Fit study was not a failure of genetics. It was a failure of choosing the wrong genes to look at to study the effects of a low-fat or low-carb diet. So I just want to go through some of the points that are often come up and explain why I think they're just completely false. And one is, some argue that single SNPs are useless. There's a trend now, at least in population genetics, to use genetic risk scores. This has evolved through the observation that looking at single SNPs does not predict complex phenotypes like heart disease, cancer, osteoporosis. And so what these population geneticists have done is say, okay, well, we're now going to look at several SNPs and try to predict more of this phenotype. And they've gone from being able to predict, 2% of the variation to say 8% of the variation. Well, that's still not really effective, because what they're not taking into account is the role of the environment, and in particular, the role of diet. So I've given a few examples in the webinar today, and others have also given some examples of specific polymorphisms, single SNPs that can predict a person's response to either caffeine or to a high protein diet. And there are a number of other examples as well. In some cases, there are examples where more than one SNP does give better predictive power. So when we look at something like iron metabolism, there are a few polymorphisms that can be used to predict something like iron overload or low iron status. Likewise, when it comes to something like gluten intolerance, there are at least 6 polymorphisms that are required to be able to predict a person's risk of gluten intolerance. But in many other cases, there are some good examples of single SNPs that have a profound functional consequence, and they can in fact be used to give dietary advice. Some argue that, well, genetics is, again, a waste of time and money. People aren't going to change their behaviors. Well, we and others have shown that they will, in fact, change their behavior if you couple that genetic information with specific actionable advice. Some argue, well, it's not ready for prime time because we need more evidence, and we need them from randomized controlled clinical trials. Well, what most of these critics fail to recognize is the vast majority of dietary advice that we give is not necessarily based on evidence from randomized controlled clinical trials. They're based on the totality of evidence. But despite that, we've actually shown using randomized controlled trials, and I've given a few examples today, that this information is in fact effective and does lead to better health outcomes. Some argue that it's all about the microbiome, and this is, I guess, a bit of a growing trend. Now, the microbiome is absolutely important. We're actually collaborating with some colleagues here in the Department of Immunology on some large-scale studies looking at the combination of the host genome, the microbiome, and other characteristics to see whether or not we can use this information to predict response. But I think the contribution of the microbiome, or at least our understanding of it, is still in its infancy. We know a lot more about the host genome. And I don't think we can say that the microbiome is responsible for everything. In fact, there's been a couple of studies now showing that the host genome, our own genetics, determines to a large extent the type of bacteria that colonize our gut. So this was demonstrated by in the UK twin study, that again, our genes determine the type of bacteria that colonize the gut. There have been some GWAS studies showing again that host genetic polymorphisms predict the bacterial composition. And just this year, another large-scale GWAS study identified genetic polymorphisms that predict the type of bacteria that colonize the gut. Now, this could explain why some studies on probiotics have not been as successful as we had hoped, because it looks like some people tend to revert to a particular microbial profile once they stop taking the specific probiotic. And this is why there's probably more of an interest now in prebiotics, because those are the foods and nutrients that actually nourish the particular bacteria, and you can actually induce certain types of bacteria, or at least create a more favorable environment. But the main take-home point that I want to make here is that our own genetics determines the type of bacteria. And so when you look at studies that link the microbiome to any kind of a response to diet, an important question to ask is it really the microbiome, or is the microbiome a predictor of the person's genetics? And it's really the host genome that is more stable and does not change that's actually predictive. Some of you might be familiar with the PREDICT trial that was published. A number of studies now, or a number of papers, I should say, have come out on the PREDICT trial. This was one from in the past year that was published in Nature Medicine, where they sought to look at a combination of factors: genetic, metabolic, microbiome in terms of the metagenome, and even meal composition. And when you look at their response, the postprandial glucose response, you can see here the number of factors predict response. And the number one factor, of course, is meal composition. And that shouldn't be surprising to anyone. But look at the second most important predictor. It's genetics. And when it comes to microbiome, it's further down the list. This is important to highlight because I think I've seen a presentation by some of the authors of this paper where they conclude that, the microbiome is really what's of greater interest, but that's not what the data shows. The data shows that genetics next to meal composition is a stronger predictor of the postprandial glucose response. They do show a similar figure when it comes to triglyceride. And in that case, genetics is not as strong a predictor when it comes to triglycerides. But part of that issue might have to do with the type of genes that have been selected. Again, just like the Diet Fit study, some of the genes that were selected are related to the steady state levels of triglyceride and not necessarily the postprandial response, like you see here with glucose. So just to go on with some of the other comments, again, by skeptics, some argue that results from genetic tests are too complex. Well, to tell someone that they're a slow metabolizer of caffeine, they need to limit their intake. That's actually quite simple. You're not giving someone a raw readout of, their whole genome sequence and trying to tell them to figure out what to do with that. That certainly would be complex, but it's important to distill that information to simple, actionable recommendations. Some argue that family history is more informative. Well, it used to be once upon a time when we did not have information about our genetics. But families, of course, share not only their genetics, but also their environments. So it's not really clear if there is something that runs in the family, if that's due to a shared environment or shared genetics. And more importantly, it's really a crude measure of a person's genetic variation. We can now pinpoint that using specific methods in genotyping. And lastly, and one of my favorites, is just follow recommendations for healthy eating. Well, if you do that for recommendations for coffee consumption, and you're part of the 50% of the population that's a slow metabolizer, then following those recommendations will increase your risk of a heart attack, hypertension, prediabetes, and a bunch of other health conditions. I think there was a press release the other day from a conference where an abstract was presented showing that coffee consumption is associated with a lower risk of all kinds of chronic diseases. To me, that kind of information or that kind of research is not really useful, because that just tells me that most of the study participants are probably fast metabolizers. And that's why it shifts the overall conclusions that a person would make if they're not taking genetics into account. So given the fact that there are some of these controversies, we decided that it was worth putting this down in a manuscript. And we just, a few months ago, Bibiana Garcia Bailo, who's one of my colleagues at the University of Toronto, and also our Director of Research and Development at Nutrigenomics, her and I published this paper in Current Opinions in Clinical Nutrition and Metabolic Care, where we highlight some of these issues that have been raised by others. And, the question of whether or not genetics contributes to individual response, whether single SNPs are of any value compared to genetic risk scores, how genetics alters behavior change, and again, the difference between the type of dietary advice that's provided. So some of those controversies are all covered in that paper. And if any of you are interested in this and you don't have access to it, you can just send me an email, which I'll provide at the end, and happy to send you a copy of this paper. So to summarize then, where are we today? Well, we need to eat. Some of you might be getting hungry right now. And we currently give dietary advice for healthy eating, but that advice is based on recommendations that I would say is based on old science from 15 to 20 years ago. So how much more evidence do we need before we can say, all right, based on a person's genotype, these are the, you should focus a little bit more on increasing this and decreasing that in terms of your specific nutrients. So is DNA-based dietary advice ready for prime time? I think if you asked me this, 10 or 15 years ago, I would have said no. The science was still in its infancy and we needed to replicate more of these findings, but I think we've come a long way. And I think the science is now quite robust, but for some markers, and I'm putting this in brackets because it's an important factor to emphasize. So not all markers are useful in terms of giving dietary advice. I haven't really gone into great detail, but some of the replication studies that I've shown were done in individuals from different ethnic backgrounds. And so if you're looking at the right functional polymorphism, it shouldn't matter what your ethnic background is. But in some cases, ethnicity is a surrogate measure of different diets and other lifestyle factors. But we and others have shown that if you're looking at the right genetic polymorphism, for example, the one in CYP1A2, if you're a carrier of the C allele, you're a slow metabolizer. Doesn't matter if you're Japanese, African, Northern European, or South American, it has the same functional effect. And it's important to recognize that. We've also shown greater compliance, and again, using randomized controlled trials. It's important, of course, that the information be actionable and personalized. Some of the earlier providers of genetic testing just gave people their genetic risk of disease, and they let them come up to their own conclusions. There's obviously greater awareness and demand for this kind of information. And we and others have also shown that this kind of information is more effective if it's given through healthcare professionals rather than direct to consumer. So we've come a long way. And again, this article in the Wall Street Journal has shown that this is no longer a concept that's futuristic. We can, in fact, give people information on their genetics and give them more precise recommendations on healthy eating. Just last year, a group of us from the American Nutrition Association put together this position paper that outlines some of the aspects of personalized nutrition and some of the definitions and identifies some of the paths towards research, education, and clinical practice. As mentioned earlier, there is one of several special issues in the journal Nutrients for those of you who are interested in submitting your work to this journal. One of them is co-edited by Dr. Lynn Ferguson and myself. And although the deadline is coming up in near the end of September, we are looking at extending this deadline perhaps towards the end of the year to enable more submissions. So if you look up under special issues, you'll find the section Nutrigenomics and the Future of Nutrition as one of the special issues. For those of you who are interested in further education, there's a course that's offered by the Dietitians of Canada on genetic testing for personalized nutrition. You don't have to be a member of the Dietitians of Canada. You don't even have to be a dietitian. And for those who are healthcare practitioners and they want to register for an account through Nutrigenomics, they'll get a discount promo code for taking the course. So right now it's around just over, I think, $130 US for this 6-hour course, but the promo code brings it down to, I think, somewhere around $70. And you get a certificate of completion, something that you can frame and put up in your office. So I'll just finish off then by thanking some of our funding partners over the years who have supported our research and the various members of my research lab and collaborators. And I just want to leave you then with this quote. Even though the term nutrigenomics and nutrigenetics are fairly new, the concept is quite ancient. And Hippocrates noted over 2,500 years ago that positive health requires a knowledge of man's primary constitution, really just an old-fashioned way of saying genotype, and the powers of various foods, both those natural to them and those resulting from human skill, as clearly personalized nutrition. So I will stop here. Thank you for your attention. Happy to take questions as part of the panel. And if I'm not able to answer any questions in the time that's allotted, feel free to email me and I will be happy to respond that way. Thank you for your attention.
Okay, thank you very much, Ahmed, for this nice and informative presentation. And now I would ask the three speakers to be connected, hopefully, and María and Alfredo, because now we will begin the question and answer session. There is a question for Anne-Marie. It says, hello, Professor Anne, I would like to ask your opinion about if it is appropriate to let the patients, knowing there are APOE genotype, taking into account that E3, E4, and E4, E4 are more susceptible for some diseases between others, dementia and Alzheimer's diseases. Okay. If it is appropriate to let the patients know they are APOE genotype, or do you think it's not? Your opinion on that?
Yeah, thanks for the question. I mean, that's a really good one. I mean, I would always say that knowledge is power if presented to the individual in the correct way. I mean, I agree, Ahmed, I really enjoyed your presentation. I agree with you that the importance of including the healthcare professional, because often when these things are, this information is given direct to consumer, people can get really stressed out if you can't put genetic information in context. Mm-hmm. But of course, there's always, you know, when it comes to APOE4, it does increase your risk of Alzheimer's disease significantly. But by knowing about that early in life and by instigating the correct behavior change, you can really mitigate that increased risk. Mm-hmm. So I think absolutely knowledge is power, and if it should be made available to individuals, but framed so that people can fully understand what genetic information means. Because in my experience, people often freak out about the fact that they might be at slightly increased risk of disease due to their genetics. Never find the fact their BMI is 40 or they smoke. And you try to explain to people, actually, the fact that you smoke or your BMI of 40 is far more consequential than perhaps your genetic information. So I think, yeah, I would say it needs to be made available to participants, but somehow it needs to be explained so it's understandable. And as Ahmed said, actionable. You know, tell people you have this genetic risk, but if you do this, you can really, really affect your health trajectory.
Yeah. Okay, thank you. That was a question, by the way, from Petros Motitis. Okay, I see here another one, which I guess, because some people didn't put the destinatary of the question, but Sruhana Mediti says, very informative presentation, interesting study on iron metabolism and PMS. That was for you, Ahmed, because I had the problem. Okay, one of my students have done a systematic review to understand the role of leptin receptor polymorphism in PCOS. Would love to hear comments on the same.
Yeah, thank you for that question. Actually, in our study, we excluded individual or women who had polycystic ovarian syndrome because we know it's a risk factor for certain premenstrual symptoms. But it's quite possible that there are some underlying mechanisms that are shared by both conditions. And also in women who have not just PMS, but PMDD, which is premenstrual dysphoric disorder, which occurs in fewer women, but causes much more severe symptoms. So again, my PhD student, Tara, is exploring other pathways, other nutrients, and other genetic polymorphisms and the leptin receptor could very well be one of the candidates that we explore. But I think it's a fascinating area because, again, as I mentioned, it's very under-researched. Funds just have not been allocated to this area in the past, but it is an area that impacts the quality of lives for really millions of women around the world.
Okay, thank you. There is another question from Paricher Amiri. Thank you for your great presentation, but I don't know who of you is the destinatary. But I think some of you have already answered this question. But does host genetic effects affect gut microbiota? I think Ahmed clearly explained this, that he will do it again. How we can use genetic and microbiota together to give dietary advice? So there are two questions. If host genetics affect microbiota.
Yeah, I'm just looking at the time that question came in. I think it was just before I showed
okay,
slides.
Sorry.
So hopefully that slide answered that. But I guess for the second part, how can we use host genetics and microbiota together? It's a great question. And I think, again, most research groups are conducting these in isolation. But the PREDICT trial really, I think to their credit, are trying to integrate this kind of information, but we have not yet seen specific examples of where the information on the microbiome can complement information on the host genome or the other way around. So I think it is something that perhaps will come in time, but we need to have that evidence, and those types of studies are only beginning to be conducted right now.
Okay, let me ask you something. And what kind of studies do we have to conduct to answer this question? To study the interaction between microbiota and host genome as a whole?
Yeah, so again, I think
artificial intelligence.
Well, it's good that you mentioned that, because I think the PREDICT trial is using artificial intelligence approaches to do that. But we hear a lot about, machine learning and AI in the field of personalized nutrition. I think in a lot of cases, it's just being used as a buzzword. When you think of exactly what artificial intelligence is, it's really just a computer doing something smarter or more efficiently than a human. And with that, you have to feed it information.
Yeah, sure.
So, an example is, something like Netflix. If you watch certain kinds of shows, it's going to recognize that, you like horror shows. So it's going to make those recommendations. But when it comes to personalized nutrition or precision nutrition, we have to look at measurable health outcomes and look to see what happens when you give people certain diets based on either their genetics, their microbiome, or the combination. What is the outcome? I think the use of artificial intelligence is really in perhaps giving recommendations for other similar types of diets, just so that there's variety in a person's meal, and you're not just saying, okay, you have to just eat broccoli. But the science is not really there yet to demonstrate, in my opinion at least, that artificial intelligence can actually give us better information or better recommendations for a person based on their genetics or microbiome.
Okay, thank you. Another question, very interesting webinar. I would like to ask if there is any ethics involved in the nutrigenomic information. Thank you. So, ethics. At the moment.
That was actually a question I was going to ask Anne-Marie regarding APOE, and I think you touched on that in terms of what people do with that information on risk of Alzheimer's and actionable advice to mitigate it.
Yeah.
I mean, I think sometimes people get a little hung up on the ethics. Genetic information is health information like any other kind of health information. And its predictive value is just like any other. I mean, I guess the benefit of genetics is that you're born with it, so you can act upon it before any detrimental effects to health occurs. But I think sometimes, for some reason, we get hung up on the ethics. I think genetic information should be treated just like any other kind of health information, rather than a separate subcategory.
Okay.
I agree 100%, and I've often said this in the past. I think the reason why some people, get freaked out about genetics is maybe based on, other types of genetics, like for high penetrance genes that predict, severe, early onset of severe diseases like Huntington's or Tay-Sachs, or, the fear that people or insurance companies or employers will use this information against you. There are certain protections and legislation that are in place, certainly in the United States, Canada, and elsewhere. But again, those refer to disease-causing genes. When it comes to the common polymorphisms that I think all three of us talked about today, it really is just another bit of health information, and not much different than your, blood glucose or your LDL cholesterol level, but it should be treated in the same manner, I think.
Okay, then there is another question that I think is for Ahmed, but you also answered this later on in your talk. What is your opinion on tests aimed directly to consumers? From Marta Alonso, the question.
I think, the reason why we chose, when we launched the company Nutrigenomics, to make it available only through healthcare professionals is, again, we weren't really interested in getting the greatest market share and getting it to as many people as possible. It was trying to do it in the most effective way, which we think is through a healthcare professional who can answer questions like the ones that, we heard today. Issues around privacy, security, confidentiality. It would be treated just like any other health information through a healthcare professional. But if you go direct to consumer, you don't know, who this company is. You don't know how they will handle this information for those who are concerned. And in some cases, companies, don't want to name names, but use genome-wide scans that can identify you. In fact, a person's identity was used to, capture a serial killer in California because one of their relatives did a 23andMe test and they had DNA from, the crime scene. Some people, it might show, impact on paternity. And there are other kinds of implications, but that is when a person uses genome-wide data or whole genome sequencing, where it can actually reveal a person's identity. It could reveal relatives. It could reveal, children that you didn't know you had, and other kinds of social issues that could be triggered by that. But when it's done through a healthcare professional, it's treated in that context of your health. And we think it's the most responsible way of providing that information.
Okay. See, this connects with another question, I think, to Professor Alfredo Martínez. Hi, Professor, may I ask your opinion on the importance of educating future medical personnel, like medical doctors and pharmacists, regarding these issues?
Thank you for the question. Indeed, this is a very important thing that Nutrigenetics and Nutrigenomics and in general omics science needs a very multilateral and universal application. Therefore, we are looking for nutritionists, pharmacists, medical doctor, genetician that can allow that in the general practice, this information can arrive to the public, to the very challenging demand that Professor El-Sohemi mentioned, and also Professor Minihane mentioned, that really this is a multidisciplinary area where it's not only research, it's not only investigations about interactions. As was mentioned, there is a role for geneticians, for medicine, for nurses, for nutritionists, for pharmacists, but also for bioinformatics. Really, the information is gathered by researchers is very important, but we need to integrate. Therefore, this is a conjoined effort with many professionals with different backgrounds in order to research, but also to implement the information is becoming more and more available through different methods and omics technologies. Thank you.
Okay, thank you. Okay, there is here a question for Ahmed. Thank you, Dr. Ahmed, for your presentation. I have a, no, excuse me, no, not this one, because that was similar to Marta's, and you have already answered this. Okay, thank you, Dr. Soemi, about the interesting presentation that you have provided us today. I would like to ask you about Mendelian randomization study design, as you show it. Can it be reliable for associations? Also, what is their main advantage and disadvantage compared to other observational studies? Thank you.
So thank you for that question. I actually just started typing a response to that question. So maybe once I respond to it verbally, I'll just put in what I've written here. And really my response is that I think Mendelian randomization can be useful when you don't have access to specific information. And in our case, we did not have access to biomarkers of iron stores. And in other studies where, again, Mendelian randomization is a predictor of exposure, like dairy intake or dietary iron, in some of these population genetic studies, they don't have information on diet. And so this serves as a surrogate measure. One of the main benefits of it is it's meant to be a predictor of long-term exposure. So again, if you have the HFE gene, you essentially have higher iron stores for life. And so any association is a predictor of exposure to higher iron stores and whatever the outcome. And likewise with the lactase gene, for lactose intolerant, it's meant to differentiate those who have low levels with high levels. But there are some limitations, and I've seen some Mendelian randomization studies that claim to link caffeine consumption to various health outcomes. But in those cases, those polymorphisms are very weakly associated with caffeine exposure. It's like, 0.25 of a cup. So less than a quarter of a cup difference. Whereas in studies where we do have information on caffeine intake, we're really comparing people who consume 1 cup versus 4 cups a day. So to try to use genetics to predict a quarter of a cup of coffee intake, when we know that other factors have a much greater impact on caffeine consumption, and then you conclude that there's no association, well, it's because your gene is not a strong predictor of that exposure. So it's important to look at each individual Mendelian randomization study separately and to ensure, A, that the gene is a strong, or the genetic variant is a strong predictor of whatever exposure is being assessed. And of course, if you do have information for the dietary exposure as well, it could complement that if you integrate that data.
Okay. Thank you. Another question. Okay, another question is about the price of genetic testing. Is for Ahmed. It seems that genetic testing, may be beneficial. Genetic testing might be beneficial for some individuals to improve their adherence to dietary advice. However, it is not cheap, I believe. So in your opinion, what kind of individuals would benefit from the genetic testing?
Yeah, I mean, I think, everyone would benefit from that. When I'm asked this question, my answer usually is, if you eat and you drink and you care about your health, then you can benefit from genetic testing. But of course, affordability and access is an important consideration. But I think the cost of genetic testing is coming down, but we need to make it more accessible, especially to vulnerable populations. But recognizing that food insecurity is also a very big factor. So even if we give genetic information for free, for a lot of people, they might not be able to afford making those dietary changes, like increasing their fruit and vegetable consumption, consuming more whole grains. So those are larger societal issues, but for those who can't afford it, anyone would benefit and everyone would benefit. You don't have to have a family history. You don't have to have an existing condition like diabetes or hypertension. A lot of people who are currently healthy want to know what they can do to stay healthy. And this is where I think there's an opportunity to learn more about your genetics.
Okay. There is another question also for you. Is there a big difference in genotype-based diet for between Caucasian and Asians?
So I didn't include it in this slide, but I used to, in my presentations on vitamin C metabolism, I show a slide where there's a polymorphism that occurs in 20% of Caucasians and 50% of East Asians. But in both cases, it's having the null genotype that makes you need more vitamin C than those who don't have it. So it impacts a greater proportion of East Asians, but you still have to know as an individual, do I have this variant or that variant? And there's many other examples of SNPs that occur more in one ethnic group than another.
Okay, that was from Anne Guyen. Another question from Francisca Serra. She congratulates every of you, and she says, research done, this is a question for all of you actually, any of you can answer it. Research done with specific nutrients as EPA, DHA, finds good relationship between metabolism, genetics, and the nutrient. However, when we talk about diet, do we already know the current composition of the foods that are available in the market? Could we rely on the food composition tables? Should we incorporate information about intake of non-nutritive items, pollutants, contaminants? Difficult question. Okay, so who is answering this question? Anne-Marie, are you answering this question?
Yeah, I mean, I think it's a good point that we don't eat individual nutrients often. We eat whole foods, and we always have to consider when we're giving dietary advice that we should be providing it on diets and food patterns rather than foods. And of course, there is a huge number of foods available on the market, and it's a huge challenge for our nutrient composition tables to keep up with the huge momentum in the field of food production. I mean, I think we do pretty well, but of course there is going to be a lag. You know, there is going to be a lag in our publicly available food composition tables versus foods appearing on the market. So I think it's a point well made. I think it's something we recognize in the field of nutrition, the challenge of nutrient composition databases and keeping those up to date. And the second point about non-nutritive items making their way into the food chain. I mean, I think that is a huge challenge to keep a check of deleterious things making their way into foods. But I guess that's kind of a challenge for the whole field of nutrition and not specifically for nutrigenetics.
Mm-hmm. Yeah. Another question for Ahmed. Has a modified methyl donor, methyl resistant diet being used for weight management? No, it's not for Ahmed. I think this one is for Alfredo, but Alfredo is not on now. Methyl donor, methyl. Oh, okay, forget that. Okay, sorry, but they moved the.
Sorry, Luisa, there was a question for me around PMS, and I typed it in, and so it's now.
Okay, that's why. Okay, that was very. Okay, so Alfredo is not now. He's not on now. I think so. Hi, Professor Anne-Marie. Thanks for your great talk. That's for you, Anne-Marie, from Anne Guyen. Do you think about time-restricted eating or fasting diet on longevity? Do epigenetic clocks play a role in longevity?
Yeah, I mean, that's an interesting one. I haven't really kept up with the field of calorie restriction and longevity. I know that the work in monkeys and apes was very, very promising, and the work in rodents was very promising. but actually it hasn't kind of translated into humans so well, is my understanding, and that you need quite severe calorie restriction over an extended period of time for it to have an effect on longevity. But certainly, I mean, there is strong sort of science that certain approaches to calorie restriction and chronic nutrition can affect metabolic profiles. But I guess Alfredo might be in a better position to answer that than me, because it's slightly removed from my interest in longevity.
Could you just simplify the question in order I can be just more precise?
Yeah, the question is if time-restricted eating or fasting diet could play a role in longevity, could help sustain longevity, I guess. And if epigenetic clocks, also about the role of epigenetic clocks in longevity.
Of course, this is a very challenging question. As you mentioned, time-restricted feeding, fasting strategies could be useful for weight management. And of course, in animal studies, there are very good evidence that show that energy restriction and fasting could help to extend longevity and reduce aging. But in humans, the evidence are less clear. In any case, as you mentioned, epigenetic could help to explain part of this result, because diet can influence telomere length, and either directly or through epigenetic processes, this a role that fasting and time-restricted feeding could help to extend life and quality of life. But of course, there is much speculation and much work need to be done in this field.
Okay, and now we are running short of time, so this is really the last question, which is addressed to all of you from Ruhana Mediti. As researchers, how do you all foresee the utilization of clinical genomics to address genetic epidemiology of various diseases? Who wants to answer this question? And Marie, are you?
Just reading the question, I'm trying to just understand the question. So it's, how do you foresee the utilization of clinical genomics to address genetic epidemiology of disease? I'm not quite sure what's meant by clinical genomics. That's where I'm kind of stumbling a little bit. I mean, yeah, I mean, I think there's role for, when we're trying to understand the relationships between diet, genetics, and well-being and disease, I think, you know, there's a huge role to play for prospective epidemiological studies and RCTs, and actually studies in clinical models or model systems. And I think it's the combination of all those methodologies will bring us to where we want to be, and no one approach is probably superior to the other. But I'm not quite sure about what's meant by clinical genomics as part of the question.
Okay, well,
Well,
I think sorry, Manet.
I just see that Sujana just added a comment by saying that next-gen sequencing and omics approaches is what she refers to clinical genomics.
Yeah.
So, I mean, I'm also still not, 100% clear on Sujana, what you're wondering, but I think in the field of genetic epidemiology, traditionally, it's just been gene disease association. And I think that's where the field has really been limiting. It's been successful for rare genetic disorders, those with high penetrance. But when it comes to the common chronic diseases, I think genetic epidemiology, gene disease association studies on their own are really, I think, a waste of time, because it's not telling us anything about the role of specific environmental factors. So whether you're using next-gen sequencing, whether you're using some other omics platform, or whether you're using a genetic risk score, I think you have to take into account the role of specific environmental factors, and namely nutrition.
Okay. Okay, thank you very much. There are a few more questions not answered yet, but Charlotte can send the question to the speaker and ask you to answer them in written form. So I think we should now close the seminar. It has been a very interesting session, and I learned a lot, and I think attendees would have also learned a lot too. So I want to finish by thanking once more the journal Nutrients for their initiative of developing scientific seminars in the web like this one, and their support in the organization, mainly logistic organization of this seminar, the speakers for their time, their availability, their willingness to participate, and their excellent presentations and their time answering questions and explaining us everything. And of course, the attendance as well, which are quite a lot actually, for your interest and your questions. And with this, I think that we can end this webinar. Okay, thank you.
Thank you, María and Luisa, for chairing the webinar. Thank you to our speakers, our attendees. As a reminder, the recording of the webinar will be made available on the webinar's webpage only after you've logged into the platform. And for those who would like a certificate of attendance, please send us an email. I wish everyone a nice day or morning or evening, wherever you are in the world. Bye-bye, everyone. Take care.
Okay, bye-bye.
Thanks, everyone.
Bye.
Bye.
Bye.